thank you for the nice introduction. And today, also thanks for the time, for the opportunity for us to present one of our study that we've been doing in Leiden University. And the study is about human-induced pluripotent stem cells reporters for high-content screening of stress response activation, identifying target organ-specific toxicity. Yeah, based on the previous study and also based off previous presentations, it's no longer questions that human bodies are most of the time exposed to xenobiotics, and unfortunately most of the xenobiotics can be quite toxic towards our body. And one of the toxicity effect that we can really see is the organ toxicity. In this rather old study from 2015, organ toxicities like the main case of drug withdrawal syndrome, drug withdrawal cases, for instance. And in this study, they showed that 21% of the drug withdrawal cases is due to hepatotoxicity. Meanwhile, for the cardiovascular toxicity and renal toxicity, there were 16 and 4% of the cases respectively. And as we know, organ toxicity is actually caused by the organ injury, and organ injury is not directly coming from the organ injury itself, but it starts from the cellular exposure towards the toxicants. And when the toxicants are exposed to, when the cells are exposed to the toxicants, the homeostasis on the cells are disturbed, and the way the cells restoring the homeostasis by activation of stress responses. And if the stress responses can help the cells to cope with the toxicant, it will regenerate and it will repair the injury. However, when the activation of these stress responses is prolonged due to the magnitude of the exposure in terms of concentration or duration, the cells will die, and then the accumulation of the dead cells will indeed deteriorate the tissue, and the deterioration of the tissue will impair the function of the organs, leading to the organ injury. And if we go back to this rather old study from 2015, it's also interesting that actually not all the organs are showing similar proportion of the toxicity. In this case, for instance, we see 21% of hepatotoxicity, meanwhile renal toxicity is rather low. So here we are suspecting there is different specificity and sensitivity of the organs towards the toxicants that are actually exposed to our body. And in order to answer these questions, in our lab, we are developing CRISPR-Cas indigenous GFP reporter cell lines in the iPSC, because we all know that iPSC is very versatile. We can use this iPSC to differentiate into organs of interest. And here, because we are focusing on the cellular level of the responses, we focus on the activation of stress responses, targeting 4 different stress responses linked to the occurrence of organ toxicity. And here, in each cellular response, we tag proteins that are associated to these cellular responses. So in this case, we have ER stress, or endoplasmic reticulum stress, with 3 different proteins, HO1, TAC1, and TRP3. We also have oxidative stress with 2 different proteins, sulforitoxin and PIR. We also have DNA damage with a well-known protein linked to this injury, P21. And at the last, we have inflammation with VCAM, ICAM, and A20 as a protein of interest for this pathway. And here, we managed to differentiate our reporter cells into 3 different lineages. We have proximal tubular-like cells, we have cardiomyocytes, and we have hepatocyte-like cells. And before we perform the biological assay, we also make sure that this differentiation, the cells upon the differentiation, they recapitulated the markers, they recapitulated the properties of the organ of interest. For instance, here we show the expression of megalin in our proximal tubular-like cells. We also show the expression of the sarcomeric protease in cardiomyocytes, and for the hepatocyte-like cells, we show also a classical hexagonal formation of the cells after 40 days of differentiation. And now we managed to differentiate our reporter cells into 3 different lineages, and then we exposed our reporter cells with 4 different reference compounds, which are the compounds that are known to induce these 4 different stress responses. So we have interferon gamma to induce inflammation, we have diethyl malate to induce oxidative stress, we use etoposide to induce DNA damage, and we also use tunicamycin to induce ER stress. And upon the exposure, we use 6 different concentration levels, and we expose our cells, our differentiated cells, for 24 hours. And then we perform live imaging with automated confocal microscopy. And then here we can also capture the expression of the proteins at the cytoplasm as well as in the nucleus. Not only that, we can also calculate the expression of this protein based on the GFP intensity, because we are using reporter cell line indeed, in order to gain the quantification and also the sense of the activation of this protein. And for the quantification, we have two different ways to express the value. We have GFP intensity normalized with min-max signals, and we also quantify the number of the positive cells that express the GFP intensity higher than certain threshold. And in this case, we use the threshold of 2 times standard deviation of the medium control. And for this case, I'd like to bring two examples of our results in cardiomyocytes and in proximal tubule-like cells. So for the cardiomyocytes, we expose our differentiated cells in these pictures with interferon gamma. We have 6 concentration level, and we measure the expression of GFP ICAM. As we can see here, from the concentration 1 until concentration 6, we see a nice increase of the GFP intensity at the cytoplasmic compartment of the cells, indicating also that we capture the correct expression of this protein in our differentiated cardiomyocytes. And then further, we calculated the number of the positive cells based on these images. And then here we can also see the increase of the number of GFP positive cells along the increase of the concentration level. Like the first concentration, which is the lowest, we see around 25% of the positive cells. Meanwhile, at the highest concentration, we captured more than 75% of the cells are committing to expressing this protein. And talking about the other proteins of interest, for instance, we also have P21, sulforedoxin, and HCO1. And these proteins are also representing the pathways that I've mentioned. And we can also see some doses of the positive controls affect the expression of this protein. And then we move forward to our proximal tubule-like cells. We expose the proximal tubule-like cells with etoposide, which is the compound inducing DNA damage. And then we measure the expression of the GFP-P21 in the nucleus. As we can see here, there is an increase of the nuclear GFP intensity upon the increase of the etoposide. Again, we also managed to capture correct expression of P21 in the nuclear compartment. And then quantifying the number of the GFP positive cells, we also see a nice dose-response relationship between GFP positive cells and etoposide concentration, where we see around 10 until 15% of the positive cells at the lowest dose, and then it keeps increasing until we see around 75% of the positive cells at the highest dose. These similar patterns also found in different proteins as well on our proximal tubule-like cells, such as ICAM1 and PIR. Now, if we are talking about comparison between organs, we try to compare the response of the ICAM1 between cardiomyocytes and proximal tubule-like cells. For instance, here we see that actually this protein is expressed quite similarly with the same concentration of interferon gamma that we use to expose the cells. However, when we use etoposide and we measure the expression of the P21 in cardiomyocyte and proximal tubules, we see striking differences between these two cell lines, indicating that proximal tubule-like cell might be more sensitive towards DNA damage based on the expression of the P21 or based on the response of this protein. So based on this preliminary data, we know that our reporter cell lines in iPSC can be differentiated into cardiomyocytes, proximal tubule-like cells, and hepatocyte. And keep in mind that these cultures are still in 2D. So now we are trying to also gain our knowledge whether or not we can also use it for 3D culturing. So for the next step, we perform the differentiation of our iPSC reported cell lines into kidney organoids using adapted Takasato protocol. So in this protocol, we cultured our cells in the 2D for 7 days, and then at day 7 until day 21, we aggregate the cells into 3D organoid. And then again, as usual, before we perform biological assay, we have to make sure that our system shows a classical expression of the organ of interest. For instance, here we generated kidney organoids, so we also have to make sure that our kidney organoids expresses the markers of nephrons. And here we showed that we have icatharine as a distal tubule marker. We also have LTL as a proximal tubule marker, and we also have NPHS1 as a glomerular marker. And as a proof of concept, we expose our kidney organoid with cisplatin, which is the chemotherapeutic compound, and we all know that cisplatin induces kidney injury in most of the patients. And here we image our kidney organoids from 0 until 48 hours live with our confocal microscopy, and then we can also see the increase of the GFPP21 after the exposure of the cisplatin. And besides cisplatin, we also use 3 different compounds, which are cephalotin, that is known to induce proximal tubules in to induce oxidative stress in the proximal tubule cells. We also have cyclosporin A, that is also known to induce ER stress in proximal tubule cells, and we also use TNF-alpha as an inflammation inducer in general. And then we can also see, for instance, for cephalotin, there is an increase of the sulforedoxin expression at 24 hours. And for the cyclosporin A, we can also capture the increase of LCN2 expression. And at the end, for the TNF-alpha, we can also see the increase of the ICAM1 expression. So based on this knowledge as well, we know that these iPSC reporter cells can also be used to differentiate into 3D test system. And yeah, to conclude this, we established a CRISPR-Cas-based GFP reporter cell lines in iPSC, which were differentiated into tissue-specific organ lineages. And our reference substances, like I mentioned before, coherently induced the GFP reporter in respective HPSC lineages. And again, the different lineages demonstrated unique concentration and temporal responses of the stress response activation. And at the end, we managed to quantify the signals based on the images using our pipeline of the image quantification. And as I mentioned before, this result is rather preliminary, so we still have a lot of things to do, and we are excited to do this. So we want to optimize the confocal imaging procedure for image analysis pipeline for the 3D organoids reporter, because imaging 3D organoids is quite challenging. And second, we also want to perform RNA sequencing to also uncover and complement the differential regulation of stress responses that we have actually shown in our reporter cell lines, but we want to know actually what happens further and deeper beyond the reporter cell line itself. And we also want to test the toxicant-specific activation of stress responses, not only the positive controls, but we also want to use the toxicants that we all know induces specific organ toxicity. And at the end, we would like to utilize this reporter panel as an advanced organ-specific in vitro test system for the regulatory purposes. And yeah, to close the presentation, I'd like to thank for all the people who worked so hard to developing this panel. Bob van der Water as the lead of our department, and I would like to thank for all of you for listening to this presentation. Thank you.
Yeah, thank you very much, Lukas. That was really a very impressive presentation. Very nice, thank you. Other questions for Lukas? Yes.
Hi, Lukas. Nice presentation. I'm just curious, when you call a cell differentiation a certain organ type, what's going on, for example, let's just take the kidney with transporters. The proximal tubule section of the kidney, a lot of drug toxicity is relevant to the transporter function. How do you deal with that, or how will you deal with that?
The question is, how can I deal with the transporter functionality in this test system? Yeah, unfortunately, we don't really test the functionality of this transporter, but we have the data of we have the transcriptomic data of these kidney organoids, for instance, or of this PTLC, for instance, and then we also compare the expression, the basal expression of this transporter among the kidney organoids, PTLC, and also the human kidney itself. And then some of the transporter, indeed, we see depletion in this in vitro test system, but the important transporter, for instance, like OCT2, which is essential for cisplatin transport, it's comparable within the test system.
Other further questions for Lukas? Yes.
Thank you for the question. So the question is, how can I quantify the image-based responses from these GFP cells? So just for the representation here, we image the cells, and we usually take 2 until 4 image points, and then from this image we segment the nuclei in order to know that, okay, there is cells, and then with the segmentation of the nuclei we perform segmentation of the cells itself, and then we define the compartment of the nuclei, the compartment of the cytoplasma, and then using this compartmental-based segmentation, we identify the response, the intensity of the GFP in this case per pixel, so pixel-based, and then we perform the summarizing of this intensity per pixel base in every cell. So the calculation is single cell based, so we can actually see the expression of this protein at the single cell level. So if you are interested more in how we calculated it, maybe we can have a discussion like off the floor, and then I can discuss it with you. Thank you.
I have a follow-up.
Yeah, sure.
So you said at the end that you're also planning to do 3D cultures, or you already did that. So how are you doing to quantify this 3D culture? Because are you like taking Z stacks and
Yeah,
just
Thank you for the question. So following up the questions from the previous, how can I quantify for the 3D structures? Yeah, it's quite tricky, because that's why in the following up the future perspective, we want to still optimize how we calculate how we quantify the 3D responses. But yeah, the Z stack is one of the way, but we can also do the Z stack projection. So we project all the 3D, we put it in one plane, and then we quantify it based on that images. But again, we miss also the layer, the expression of the protein in every layer of the cells. But it's also a bit like a balance. You want to quantify it like structure-wise, or you want to quantify it in population-wise. Like you can just quantify it with green signals compared to the nuclear signal, and then you get the expression of the green. Or you really want to have it like single cells. That's like, we need to wait at this point. Thank you so much.
If there are no further questions to Lukas, I have one. I would like to know how you choose your iPSCs, your donor cells, and if you experience, I'm sure you do, and how you deal with donor-specific differences.
Yeah. Thank you so much. So the question is already mentioned. So in these reported cell lines, we use one cell lines. We use sigma cell lines, if you are familiar with this. But again, because we generated multiple reporter cell lines here, we have actually 14 different reporter cell lines, and even with one donor cell lines, we still see interdifferences of the reporters. So yeah, at this moment, we are collecting the samples for the RNA sequencing for every single reporter cell lines, and we also have different donor as well. And until now, we managed to differentiate them into the iPSC, into the organ of interest, and then we also collected the baseline transcriptomic signals of this, and then we would like to compare based on this transcriptomic level. But so far, like immunostaining-wise, marker genes expression-wise, or protein expression-wise, they are comparable.
Okay, so thank you very much, Lukas, again.